All right. This video is going to be a short one, and it's focused in on this. And so at the end of the last video about atoms, we started to build molecules. And remember, molecules are atoms joined together by covalent bonds. And we have these two types of bonds. And they are different because the atoms joined together have either the comparable electronegativity or different electronegativity. And you don't need to calculate or know the electronegativity. Again, we just have four elements that we're really interested in, and what we're just going to point out right now is that these guys are on the same row, and so oxygen is going to have more electronegativity than nitrogen and more than carbon. And then it's a little bit hard to judge because they're on different rows which of these has more electronegativity. But it turns out that hydrogen's electronegativity is similar to carbon's. Carbon's got more proton pull, but the outer valence electrons are further from the nucleus. And so about the same, and that means that hydrogen is less electronegative than oxygen. And so what we'll see is that water forms polar covalent bonds. And then we already talked about, we saw in chapter 2, a simple molecule like methane is nonpolar. And it turns out that these types of connections are present in a lot of fats and oils. And the entire reason that a jar of dressing separates into fats and oils is because the fats and oils get pushed out of the water because the water can't interact with other water molecules as a result of this slight charge that we talked about in the previous video. And so water is essentially sort of dancing with the other water molecules, and it can't do that with the fats and oils, the carbon-hydrogen bonds in fats and oils, and so these get pushed out as oil droplets get excluded, like that. And that's the basis for a lot of the chemistry and biology, is that some molecules are polar, some are nonpolar. A great big molecule like insulin has lots of surface on it, and some of the surface is polar and some is nonpolar. As a result, the polar surfaces of insulin are able to get dissolved in water, dance with water, and the nonpolar surfaces are not. Overall, insulin dissolves well in the blood. So overall, it's carried through the blood as a dissolved molecule, like that. And so we're going to build bigger molecules in this water environment. Again, big theme, water environment. And water is polar, so that's what's going on there. Now, here's another thing, is that when table salt dissolves, sodium and chlorine are charged, and they dissolve really well in water. That's why the table salt separates. Is that the oxygen in water molecules is attracted, because it has a slight negative charge, attracted to that sodium and is surrounding it. There's said to be a shell of hydration around that sodium. And there's a shell of hydration around the chlorine too. But in this case, it's the hydrogens that are especially attracted to the chlorine, like that. And you know that when there are a couple things. For example, when a solution of salt water is cooled down, eventually the salt might precipitate. So that's an interesting chemical phenomenon we'll just talk a little bit about. This water is kind of dancing around. It's interacting with different molecules in the water and dissolving them. And this dancing around is how the water holds heat. So heat in an object is held by atomic and molecular vibration, and as the heat goes up, there's more energy held, and that water is dancing around more vigorously, and the warmer water can dissolve salts or insulin, molecules better. We can cool it down, and eventually the water is moving so slowly it can't really dissolve it. These things precipitate out. So it's one idea. And another idea is that if you keep adding this, eventually it won't be able to add. You won't be able to dissolve more, right? You can saturate the ability of the water to dissolve this stuff. And so there's very interesting qualities about life and water, and we're just starting to introduce them. But again, we introduce a set of principles, and then it's just more of those principles. Like I already pointed out, insulin's really big and has a lot of complexity to it, but it's a repeating features of covalent and non-covalent bonds because there are carbon-hydrogen bonds and oxygen-hydrogen bonds and so forth and so on throughout that molecule, like that. Okay, so that's that. Here's just, this is hydrogen gas. So in the atmosphere there's some hydrogen and it exists as a molecule, right? That's a molecule because it's joined together by a covalent bond. And in this case, these would be non-polar because they're two hydrogens, so they have the same electronegativity, like that. So a non-polar covalent bond and then polar. What's the difference is, in this case, hydrogen sharing with hydrogen, same electronegativity, sharing with oxygen, different electronegativity. And here's a really nice image where it shows this range of differences in electronegativity between two interacting atoms. And we've already talked about the extremes. Here's hydrogen, no difference in electronegativity, so non-polar. And at the other extreme, we have a big difference in electronegativity. Remember, sodium and chlorine are on the same row, but chlorine has like 6 more protons pulling. So big difference in electronegativity. And in this case, they aren't even going to share the electron. Chlorine is just going to take it. Like that. So see, that's a nice way to see that feeling of like, yeah, we can share these electrons, but if our electronegativity is really different, I'll just take it. And see, water is leading out this way, moving out in this direction, quite a big difference in electronegativity, but still sharing. There's nitrogen, right, on the periodic table, a little bit less electronegativity, carbon even less, and carbon has comparable electronegativity to hydrogen, so we have this nice spectrum of range. It's nice to see this because it turns out that water is close to this, and in water, if you just had water molecule by itself like that, it would be very stable. These atoms are most stable when their outer valence orbitals are full, so very stable. However, when you add water to water, this is called a non-covalent bond because the electrons are not being shared, but there's some attraction. So maybe we'll just make it a little different color here. There's some attraction there, maybe a dotted line, like that. And it's less stable, so this water molecule goes spinning off, and another water will take its place. But while it's there, this oxygen's bonds to these guys are weakened because there's an alternative possibility, and sometimes oxygen will make a covalent bond with a neighbor. So the covalent bonds will shift like that because they're weakened in that environment. And so it's interesting to see that when something's dissolved in water, the water is actually weakening the environment of the material that's dissolved. You'll see a medicine, and it will say on the medicine, on the pills, it will say, store in a cool, dry place. When you take the medicine, right, the pill dissolves in the liquid water of your body. But if you want to store it and keep it and have a long shelf life, it's good to keep it cool. Why? Just because, again, heat is a way that molecules and atoms hold energy. And, warmer is more active, more likely prone to break the chemical down, the medicine down. And then dry, because of what we're talking about here. When the molecule gets dissolved in water, it destabilizes it. Part of being dissolved is, part of the process of being dissolved kind of destabilize it because it gives binding alternatives like that. We should just say tier 2 that it's good. In our course, we're going to define in chapter 8 and 9, so in just a couple weeks away, we're going to talk about chemical reactions. And a chemical reaction is going to be making and breaking covalent bonds. And so we usually think and appropriately think of that as being part of metabolism. Digesting food is making and breaking covalent bonds, releasing food energy like that. But we're claiming that the water is constantly going through some chemical reactions too, because this molecule will form a hydrogen bond. The hydrogen will form a covalent bond with this oxygen, and as a result, oxygen will lose a covalent bond, so it'll make a covalent bond and lose one, and that's happening all the time, very, very fast. And that's going to be another mini lecture about pH, because pH is a measure of how much of that making and breaking of covalent bonds is happening. Water can do this all day. So it can just be sitting there making and breaking covalent bonds very rapidly all day, because this making and breaking of covalent bonds is a way that the water is just simply holding the heat. It's not gaining energy or losing energy, it's just bouncing these guys back and forth around like that. Water molecules are vibrating and wiggling, and that includes making and breaking covalent bonds. It takes energy to make and break covalent bonds, but it's all balanced out. Good. Okay, cool. So, there's the book just showing you the gaining and loss in sodium chloride of the electron, like we've been talking about. And then here in the handout, it talks about non-covalent interactions. And biologists and chemists really typically call these bonds, and that's just too bad. It's too bad because we have covalent bonds, and those are the sharing of electrons. And non-covalent bonds or interactions occur between molecular atoms. And, it's describing that a little bit here. It's too bad they're called bonds because they get confused with covalent bonds. Non-covalent bonds or interactions, there is no sharing of electrons. So covalent bonds sharing electrons, non-covalent bonds not sharing electrons. And there are some different types of non-covalent interactions, and one is this dipole-dipole interaction, which is happening in water, between water. So between water, there can be a non-covalent bond between hydrogen and oxygen. Not only is that it's too bad that it's called a non-covalent bond, it's also too bad that it has a special name, it's given a special name, that's called a hydrogen bond. So water has lots of hydrogen bonds, they're very dynamic, unstable, weak interactions, constantly being made and broken. Because they are non-covalent, they don't involve sharing electrons, like that. And we talked some about ionic interactions. That's the charge difference between the ions in sodium chloride. And we'll talk about some London dispersion forces when we get to talking about the molecules in the cell membrane. And we're getting close to that. Okay, so just more from your handout about polar and nonpolar. There's water. There's a hydrogen bond from chapter 2 or 3. 3 now, like that. Hydrogen bonds in water, dynamic and weak. And Molecules that can form hydrogen bonds are called hydrophilic, and those that can't are called hydrophobic. So oil, fats, carbon-hydrogen bonds are said to be hydrophobic, and we use that term a lot. Part of the surface of insulin is hydrophobic, and part is hydrophilic, like that. There's that again for some reason, and just showing the salt getting dissolved in water, like that. And the next thing to talk a little bit about is molarity. And actually what I'll do is I'll stop this lecture and make another one that includes molarity and pH.